Good evening. In today's lecture, we will be discussing questions related to the lectures covered in week 6 of the NPTEL course, Pharmacognosy and Metabolic Engineering. The first question is, strong activation of CrGATA1 takes place in the presence of light. Based on this statement, identify the false option from the following. Option A, strong activation of CrGATA suppresses the light-sensitive genes. Option B, CRPIF1, a transcription factor, degrades in the presence of light. Option C, strong activation of CrGATA1 enhances windowlin formation. Option D, the presence of CRPIF1 influences the activation of CrGATA1. You can take a couple of minutes to think about this question. Okay, so we'll break down this question into parts. First, we need to know what CRGATA1 is. Then we need to know where it is involved. Then we look at the options. So, shown here is a schematic representing different transcription factors or transcription activators that would allow for expression of gene with respect to various stimuli. What we are interested in is the CRGATA1. So CR here stands for Catharanthus roseus. Now, this in combination with the CRPIF1, both of these are essential for production of windowlin and hence windblastin and windcrystal. The images here are taken from this particular article where the effect of light and how it can regulate the windowlin pathway is discussed. So, if you see here, what is shown is in the presence of light, CRGATA1 is activated whereas CRPIF1 is suppressed. If CRPIF1 is produced, that would suppress CRGATA1 activity. So, how you can understand this is when light is there, the CRGATA1 is produced and it would upregulate T16H2, T3O, T3R, D4H, and DAT. And the respective reactions that are catalyzed by these genes are shown here, from Tabor-Sonin to windowlin. So, when light is present, you get windowlin. When light is absent, CRPIF1 would suppress this CRGATA1. And so there is nothing to activate these genes. Also, the CRPIF1 in turn suppresses the expression of these genes. So windowlin will not be produced. So this is the basic idea. Now let's go to the question. So we are asked for the false option here. So strong activation takes place in the presence of light. Based on this, identify the false option. The strong activation of CRGATA1 suppresses the light sensitive genes. CRPIF1, a transcription factor, degrades in the presence of light. Strong activation of CRGATA1 enhances windowlin formation. The presence of CRPIF1 influences the activation of CRGATA1. Now, we saw that in the presence of light, CRPIF1 cannot be active. So option B is correct. And in the presence of light, CrGATA1 is active, so windowland will be produced. So option C is also correct. The fourth option D. It says that the presence of CRPAF1 influences the activation of CRGATA1. So here, if CRPAF1 is present, it is shown that it can repress CRGATA1. So that influences the activity of CRGATA1. So that is also correct. But option A, which says that strong activation of CRGATA1 suppresses the light sensitive genes. What we observe here is that it's an activator. It is never shown to be a repressor. So option A is the wrong answer. That's what I have typed here. And it's very clear that transcription factors are activators rather than suppressors. So only if it's a repressor, it can suppress the effect of particular genes. The next question is, CR-MYC1 is expressed in co-transformed hairy root cultures of C. roseus. What was the effect of this transformation on the levels of terpenoid indole alkaloid biosynthesis? Option A, ajmalicin and catharanthin content increased equally. Option B, ajmalicin and catharanthin content decreased. Option C, catharanthin content increased more than ajmalicin. And option D, ajmalicin content increased more than catharanthin. Okay. Again, this is a question on the effect of expression of a particular gene. Here, this is called CR-MYC1. CR again is from Catharanthus roseus. You can take a couple of minutes to think about this question. Then we will look at data from a paper which, where this experiment was performed and the results of that experiment. Hi, I have started with the lecture. Can you hear me? Can you hear me? So the question we are dealing with here is the effect of expression of CR-MYC1 on terpenoid indole alkaloid biosynthesis. First, let us look at the pathway. Again, the link to this paper is given here. So, this pathway begins with tryptophan, and you also need cephaloganin. So, these are you have the precursors from two different independent pathways. One is the shikimate pathway, which gives you tryptophan, which gives you tryptamine indeed, and the other is the methyl erythritol phosphate pathway, which gives you cephaloganin. Now, both of these combine to form streptozidin, and from streptozidin you get catharanthin, and then finally vinblastine. Now, when CR-MYC1 is overexpressed, what was done in this particular paper is that They have calculated both the levels of mRNA that are being produced when this CRMIC1 is being expressed, and the effect of a gene, which is SGD, which acts on this strictocidin. So this particular experiment was done in more than one particular cell line or plant cell line. Here you see that it's a transgenic MY3 and MY1 lines. So this was done in order to see if there is any effect of transgenic cell line specific changes. So they wanted to negate that independent of whichever plant line you take, you still get the same result. That's why this was done on two different lines. And the control here was this lysogenes, ATCC 15834 strain. So the first thing that they did here was they had introduced this CRMIC1 in these two and then checked for their relative expression levels. So with respect to the control, the CRMIC1 was overexpressed in both these strains with significant statistical significance. Then, in order to check how this has an effect on the expression of genes, this one gene, SGD, which we had discussed earlier, which is a glucoside uh, a gene for this strictocidin. So they observed that this gene is also overexpressed. The more that CRMIC1 is getting expressed, proportionately, mRNA levels for SGD was also increasing in both the cell line. Once this was done, they had also measured how much of each of these metabolites, ajmalasin and catharanthin, was increased. And what they could observe is that ajmalasin here could be increased, and in catharanthin, in one particular cell line, they saw a decrease in the amount of catharanthin. And here they show that although the CRMIC1 overexpression appear to increase the levels of both levels of ajmalasin than catharanthin, the effect of this accumulation on different TAs differed. They say that catharanthin showed a maximum of 4-fold increase, whereas ajmalasin was altered up to 14-fold in the transgenic lines versus control. Hmm. This result suggests that the metabolite flux changes due to CRMIC1 overexpression were more to the advantage of ajmalasin than catharanthin. So as you see here, compared to the control in both the lines, transgenic cell lines, you observe ajmalasin being overexpressed, ajmalasin being accumulated a lot. But with respect to catharanthin, it's not, uh, it changes from cell line to cell line. So because of this reason, we would go with this option. Ajmalasin content increased more than catharanthin, which was what was observed here as well. Right. The next question is match the steps of terpenoid indole biosynthetic pathway with the enzymes that catalyze it. There are four different steps. Des-acetoxywindolin to D-acetylwindolin, tabersonin to loschnericin, tryptophan to tryptamine, D-acetylwindolin to windolin. And four different enzymes are given here. So you can take a couple of minutes, think about the pathway and where it starts. For example, like we saw in the earlier slides, tryptophan to tryptamine is the first step that requires tryptophan decarboxylase. So keeping that in mind, we will further go with the question. You can take a couple of minutes and then try to answer this question. Okay, so if we look at this pathway, it begins with tryptophan to tryptamine that's being decarboxylated. So for this, you need this enzyme TDC. So 3D. This gives this lets us to choose either option B or option C. Now we look at the pathway once. We see that tryptophan to tryptamine is TDC, tryptamine to strictocerone is SS, and you have here all the way from taberzonin to desacetoxywindolene. Now from des-acetoxyvindoline to D-acetylvindoline, which is a typical hydrogenase reaction, which needs alpha ketoglutarate that gets converted to succinate and carbon dioxide. You need a des-acetoxyvindoline 4-hydroxylase. And we see here at the end of this reaction, here where I'm pointing at, there's an OH group. So this is the chemical conversion. So this reaction needs D4H. Then from D-acetyl vindoline, you get vindoline. So here what's happening is this hydroxyl group now is an acetyl group. So this acetyl CoA, which is a cofactor, provides an acetate, and that is being covalently linked here. So this is an acetylation reaction. Now if we look at the options here, option 4, D-acetylvindoline to vindoline needs DAT. So 4C, 3D, and des-acetoxyvindoline to D-acetylvindoline is D4H, which is a hydrogenase reaction. So 1B, 2A, 3D, and 4C. Option C is the right answer here for this question. Ha. The next question is, in a metabolic engineering approach, scientists overexpressed geraniol 10-hydroxylase (G10H) and octodecanoid responsive Catharanthus AP2 domain protein, which is the ORCA3. We were discussing about this particular transcription factor earlier in the hairy roots of Catharanthus roseus that resulted in higher concentrations of catharanthus. Upon eliciting the co-expressed hairy roots with a phytohormone, accumulation of catharanthin reached 1.96 mg per gram dry weight. Identify the classical phytohormone employed to induce catharanthin in hairy root. Four different options are given here: abscisic acid, triiodobenzoic acid, jasmonic acid, and phytosulfoquene. You can take a couple of minutes and then think about what would be this classical phytohormone to induce catharanthin in hairy roots. Okay. So what they say is overexpression of G10H and orca3, both of which we know are involved in terpenoid indole alkaloid biosynthesis. Huh. Can you hear me, Swati?
Yeah, good evening, sir.
Good evening. Yeah. So I have started going through the lecture. There were people coming in and going. We can restart it, not an issue. Right. So the first question that we are looking at for this week is strong activation of CRGATA1 takes place in the presence of light. Based on this statement, identify the false option from the following. Four options are given here. Strong activation of CRGATA1 suppresses the light sensitive genes. CRPIF1, a transcription factor, degrades in the presence of light. Strong activation of CRGATA1 enhances windowland formation. And the fourth option is the presence of CRPIF1 influences the activation of CRGATA1. You can take a couple of minutes and think through this question and then unmute. Right. Yeah, you can unmute.
Um, I think the false statement we have to tell now.
Identify. Yeah.
Strong activation of CRGA1 TA1 takes place in the presence of light. So I think first one is the false option because option D
Uh-huh.
CRPIF1
IF1.
transcription factor degrades in the presence of light. That is um, because CRP1 F1 is activated in the dark.
Yeah, correct.
Strong activation of CRGAT1 enhances windowland formation. CRGATA1
Hmm.
enhances windowland formation in the presence of light.
Yes. CRGATA1 gets activated in the presence of light, and CRPAF1,
And
it's a
forms the
repressor.
windowland.
Huh.
Yeah, the presence of CRP1 influences the activation of CRGATA1. I think option A is the false.
Right. Yeah. So this is a schematic from a paper. You can check this later. So here they mention how CRGATA1 is activated in light and CRPAF1 is activated in the darkness. And how CRPAF1, it's a repressor. It will not allow CRGATA1 to function. So if CRPAF1 is present, we will not get Windolin. But if CRGATA1 is present, we will get Windolin. And like you mentioned, strong activation of CRGATA1 suppresses light sensitive genes. So because CRGATA1 is a transcription activator, and it increases the expression of genes, there is no documented role of this in the suppression of genes. So that is the false option, like you had mentioned. Right. The next question is CRMYC1 is expressed, CRMYC1 is expressed in co-transformed hairy root cultures of Catharanthus roseus. What was the effect of this transformation on the levels of terpenoid indole alkaloid biosynthesis? And four options are given here. Ajmalsin and catharanthin content increased equally. Ajmalsin catharanthin content decreased. Catharanthin content increased more than ajmalsin. Ajmalsin content increased more than catharanthin. You can take a couple of minutes and then think about this question. So here again, this is directly given from a paper. We will discuss the experiment that was done in the paper to confirm this, and then look at the right answer. You can take a couple of minutes. Yes, you can unmute.
Um, I think this CRMYC1 is
Hmm.
expressed in structure. It is to enhance the content of the alkaloid, the effect of transformation on the levels of terpenoid indole biosynthesis. I think it is um, Ajmalis option D, content increased 13 to 14 fold more
Hmm.
than catharanthin.
Okay, but do you know what gene is being overexpressed when CRMIC1 is being expressed in this hairy root cultures? It should give rise to overexpression of some gene, right?
Yeah.
Huh.
Uh,
So do you
uh,
know which gene from the pathway is being overexpressed?
cDNA to the in rhizogenes, which is which gene? Some what the name of the gene?
No issues. So this is the pathway. Here we have cyclosidin, which is a glycoside. From cyclosidin, when you have when it leads to the production of other compounds like ajmalsin and so on, this glycoside is being removed, meaning this glucose moiety is removed. So what these authors had done in this paper, again the link for this paper is given here. What these authors had done in this paper is they had taken two different transgenic lines, MY3 and MY1, because they wanted to see when you introduce the CRMIC1 into a particular system, it should have a similar effect independent of the nature of the system. So they had taken both these two transgenic lines, added this CRMIC1, and they saw the expression levels when it is being overexpressed. So compared to the control, which is an empty vector, they saw an increase in CRMIC1 levels. Then they also looked at one specific gene, which is SGD, which removes this glycosyl moiety from strictocidin. And they observed that there is an overexpression of this gene as well. Then, to see the effect of the overexpression of this particular gene, SGD, they measured the accumulation of both ajmalacin and catharanthine. What they observed is when they looked at ajmalacin, it had similar overexpression levels, but catharanthine had uh, uh, an increasing overall trend, but the levels of increase were not the same. And if they compared this with respect to the control, like you had mentioned, ajmalacin was altered up to 14 folds, whereas catharanthine showed only 4-fold increase. So with this, they were able to say that ajmalacin content was the one that had a higher increase compared to catharanthine. You are right about the option. It's just that we need to also know why the CRMIC1 introduction is altering ajmalacin expression. So that now we know that introducing CRMIC1 is altering SGD. That is why we have an increase in ajmalacin levels. I hope that was clear. Okay. So the next question is match the steps of terpenoid indole biosynthetic pathway with the enzymes that catalyze it. And four different reactions are given here. Desacetoxyvindoline to deacetylvindoline, tabersonin to loschnericin, tryptophan to tryptamine, and deacetylvindoline to vindoline. So you can take a couple of minutes, think about what reaction is being catalyzed here, and then maybe come up with the right answer here. Okay. Yeah. So, do you want to try answering this question? Okay, so this is again simple. You have also come across these reactions. Huh. Yeah. You
Sir,
yeah.
tryptophan to tryptamine.
Hmm.
Third, what is the TDC? D option.
Hmm. Yeah. It's a decarboxylation reaction. So you have tryptophan decarboxylase. Yeah. So that gives us either option B or option C now. Hmm.
Diacetyl indolent to the tabersonin to locnaricin, is it T67E?
Yes. In both options, it's T670. So yeah,
Oh, sorry,
that is confirmed. Yeah.
I didn't see here.
Right.
Even third is D. Third is D.
Yeah.
Diacetyl indolent.
You can try
Confused
to.
with D.
Okay. So do you know what is the chemical difference between diacetyl and vindoline structurally?
Deacetylation.
Structurally?
No, no, no. Deacetylation reaction.
No, no. So we get vindoline from diacetyl. Only if you have the reverse reaction, that's a deacetylation reaction. So the forward reaction is an acetylation reaction or an acetyl transferase reaction. So structurally, if you see, diacetyl has an OH group here, which is getting replaced to acetoxy here, an acetyl group. So an acetyl group is added here, which needs an acetyl transferase protein or an enzyme, a DAT enzyme. So for that particular reaction, which is reaction 4, you need DAT, which leads us to option C. So because you're familiar with the other reactions, I'll also cover one more reaction here, the only unleft reaction, which is des-acetoxy-vindoline to D-acetyl-vindoline. And here, if you see, it's an enzyme called D4H, which is des-acetoxy-vindoline 4 hydroxylase. So what is happening here is this group of enzymes, hydroxylases, they all take in alpha ketoglutarate and iron as a substrate, a cofactor, convert it into succinate and carbon dioxide, and add a hydroxyl group here to the enzyme at the substrate at the fourth position. So that's why this is a D4H enzyme. So that leads us to option C. Hmm, right. The next question is, in a metabolic engineering approach, scientists overexpressed geraniol 10 hydroxylase G10H an octadecanoid responsive Catharanthus AP2 domain protein, or CAR3, in hairy roots of Catharanthus roseus that resulted in higher concentrations of catharanthin. Upon eliciting the co-expressed hairy roots with the phytohormone, accumulation of catharanthin reached 1.96 mg per gram dry weight. Identify the classical phytohormone employed to induce catharanthin in hairy roots. Yeah, and four options are given here: abscisic acid, triiodobenzoic acid, jasmonic acid, and phytosulfokyne. So you can take a couple of minutes to think about what would lead, which of these is important for catharanthin production, and then we'll discuss that further. Yeah, so here we are looking at the effect of a particular phytohormone in increasing catharanthin levels. So, which one do you think here could be the possible phytohormone?
I'm not aware of this question.
Okay,
And,
no issues. Do you think, okay, do you know about abscisic acid?
yeah, vitamin C.
That's ascorbic acid.
Yeah, yeah,
Haha.
uh,
So this is a growth hormone in plants, like indolestic acid and
yeah,
whatever.
phytohormones in plants. Yes.
Yeah. So do you think that will be involved in inducing catharanthin?
One second.
Sure, sure.
Yes, sir.
Do you think abscisic acid would be involved in catharanthin expression or stress response or growth?
Growth is a phytohormone.
Huh.
Um, so responsible, I think.
So it cannot be responsible. What about triiodobenzoic acid? Again, that's also a
Um,
plant growth regulator.
Jasmoic acid.
Yeah. Here, correspondingly, jasmonic acid is the phytohormone because that we have also seen in questions in the earlier lectures that it's an elicitor for secondary metabolite production, including catharanthin and windolin. So that is the specific phytohormone that we are looking for here in this question. Right. The next question is, what is the relation between windolin accumulation in latifier cells and leaf expansion and Catharanthus roseus? And the options are windolin accumulation decreases as the leaf expands. Windolin accumulation gets inhibited as the leaf expands. Windolin accumulation increases as the leaf expands. There is no change in windolin accumulation with respect to leaf expansion. So you can think about where all this process happens, which is the process of production of windolin. So windolin requires multiple reactions and not all the reactions happen in the same cytosol. It happens in multiple different compartments and that in a way has an effect on the overall metabolism of the cell. So you can think in that particular aspect and try to combine that with expansion or contraction of the leaf. Think about this for a couple of minutes and then we'll discuss this question. Right. So, to start with for this question, think about the initial steps of this particular process. Where do you think this happens? The initial step where windowlin is being produced. Okay. So if you think about this particular process, so the first initial steps for indolent biosynthesis, it happens in epidermal cells of the young leaves, like we saw earlier of these reactions. But the later steps, it happens in this lattice-fire and idioblasts. Now, windolin finally accumulates in the vacuole of idioblast and lattice-fire. Now, one might ask why this accumulation happens majorly in the expanding leaves. Now, that is because these young leaves, they do not have the specialized lattice-fire or idioblast that is needed for the final enzymatic steps. Only if that environment is present, these reactions would happen. So, in order for that environment to be even able to present, the leaf has to mature and expand. So, that is what is limiting in this process. So, that is why it's a positive correlation or a proportionate thing. As and when the leaf expands, windolin accumulation would increase. So, that is why option C is the right answer here. Was it clear?
Yes, sir. Yes, sir.
Right, right.
Absolutely. Yeah.
Okay, right. The next question is select the correct transporter protein that transports catharantin into cell surface. Four options are given here: NPF transporter, ABC transporter, antiporter, and TPT2 transporter. This again is an interesting question that was based on a paper published in, I think, PNAS. We have the data here, we'll discuss that. So you can take a couple of minutes, think about this question, and then we'll discuss it. Yeah, you can unmute.
I think it is the option B, ABC transporter,
Hmm.
and uh, ABC transporter exports the cathepsin thing into the cell
Rightly.
because NPF transporter exports the MIA from
Yeah, I
the
think I lost
vacuole.
you. Can you repeat? I think I lost you. What from vacuoles?
I think option B is the correct ABC transporter.
Huh.
Transports cathepsin thing into the cell.
Okay.
NPF, NPF transporters, it exports central MIA intermediate
Oh,
vacuole.
okay. Okay.
And uh, uh, and about the antiporter, it is transports the windolin and cathepsin.
Hmm.
So I think option B, ABC transport.
Okay. So this is an interesting question. So here they had done a couple of experiments. So what they had done is they had taken an yeast strain with with the major ABC transporters being knocked out, and they made the yeast strains to produce catharanthin. And after making it to produce catharanthin, they did a couple of changes. One, these ABC transporters need ATP for their functioning. So they compared the levels of catharanthin within the cells after making these mutations in such a way that this ABC transporter will not be able to bind to ATP. So basically, it will be accumulated within the cell. And they saw a difference comparing the ones that can bind ATP and the ones that cannot bind ATP. With which they were able to say that in the ones which cannot bind to ATP, you have more of catharanthin being accumulated within the cell. Whereas in the ones that can bind ATP, catharanthin got secreted. So with this, they were able to say that it is indeed that these ABC transporters are the ones that are responsible for accumulation of catharanthin or transport. So that's why it's ABC transporter here. The next question is to enhance the production of indole alkaloids in Catharanthus roseus, hairy root cultures were established, and a transcription factor gene from C. roseus was overexpressed in the root culture. The chosen transcription factor for this metabolic engineering approaches and four options are given here: CRMYB1, CRMIC1, CRWRKY1, CRWRKY11. So you can take a couple of minutes, think about this question, and then we will look at the role of one of the specific transcription factors. Right. So even earlier in one of the questions we were discussing this. So, well, Swati, what do you think would be the right answer for this?
Is it option B?
Yeah. Do you know about the other options by any chance?
No.
Okay. So here, MIC1 is like we know that it's responsible for production of these indole alkaloids. MYB1 is a part of those that produce flavonoids. That is again another class of transcription factors. But yeah, what we wanted was for this indole alkaloid, so CRMIC1 is the right answer. And WRKY, it's involved in plant defense. So is the case with this WRKY11. Again, but people say that it has been reported to participate in this jasmonate signaling and plant defense. But it's only in jasmonate signaling and not, it would not activate indole alkaloid pathway. So with that, we will say that MIC1 should be the right option. The next question is match the enzymes on terpenoid indole alkaloid biosynthesis on left hand side with the appropriate cellular locale on right hand side. We have T16H2, 16-OMT, N-methyl transferase, and D-acetoxyvindoline 4-O acetyl transferase. And so it's actually here cytosol, external phase of ER, vacuole, and chloroplast. Uh, to, to think about this question, I would suggest you which of these is the cytochrome P450 enzyme. Think about that because cytochrome P450 enzymes require a specialized environment. They are membrane proteins. To start with, you can think about this problem that way. You can take a couple of minutes to think about it and then we will look at the right answer. Yeah, you can unmute. So when you're trying to answer this question, you can uh, justify why a particular enzyme here would require a particular organelle. Okay.
Yeah, the cytochrome P450 enzyme that
Hmm.
is the Vindolen oxidase.
Hydroxylase, T16H2, right?
It is this that. So is it option D? Deforest and external protein.
Um, okay, you can tell me why do you think it's option B?
Option D, D.
D. Why do you think it's option D? Yeah, you can tell me why, like whatever reason it is, you can tell me.
D4H is there in the external face of endoplasmic reticulum. That's not
What is
sure.
D4H? What is the reaction happening here? We just looked at it. It's actually not D4H. So I think there is an error in how the question was made. It's actually DAT. This stands for DAT, whereas D4H is the other enzyme that's prior to this.
D4H is deacetovindolen to deacetylvindolen, and then deacetylvindolen to vindolen with the DAT.
Vanilla. But what is the environment do you think it's needed? Do you think you need it to be on the external face of ear?
Like idioblast lenticular it requires.
No, no, that is the tissue. Within the tissue, you have these organelles, and each organelle has a specific chemical nature where these enzymes can function. This chemical
Okay, okay.
nature, yeah, yeah.
Is it will be located in the cytosol?
Which one? Option D. I mean, sorry, this D4H or
Means
DAT. That's what you're
D4H.
saying.
Yeah,
Huh.
yeah.
It can function in cytosol because the reaction
Cyt-
that it's catalyzing is very simple. Right. That is cytosol.
Internal tissue.
Can you repeat? Can you repeat? I think I lost you.
Internal tissue, I think. That's
Okay, okay.
right.
No issues, no issues. You can continue. You were telling option D, D4H or DAT. Either of it works in cytosol because the nature of the reaction is simple. Right.
Yes.
Huh. What else?
Um, T6, this one T16H2
Hmm.
is the option A. In the external phase of
Huh. Okay. Let's say it's in the external phase of ER, but do you think it would
uh,
necessarily require external phase of ER? Is there any requirement?
I don't know.
Okay. So there is one way to think about this. So this, I was asking earlier at the end of when I started this question about cytochrome P450 enzymes. Do you know about cytochrome P450 enzymes?
Cytochrome P450, I told this Dabersonian T16H2
Hmm. So
Kilo.
these cytochrome P450 enzymes are integral membrane binding proteins, meaning these function by binding to a membrane. So they require lipid bilayer for folding and its functionality. So they require a membrane for its functionality. Now, an additional requirement is that they need electrons from NADPH to catalyze the oxidation, which is why here you need this phase of ER, which has a lipid membrane for T16H2 to function. And other than that, you have NMT. So NMT is N-methyl transferase. Now, What was done in one particular study is they compared the sequences of N-methyl transferase from Catharanthus with other sequences from other sequences of methyl transferases. And they observed that these methyl transferases, whatever sequences, according to phylogeny and evolutionary history, this N-methyl transferase could have originated from a particular enzyme that was initially present in chloroplast. And there is also this one study where they say that N-methyl transferase was observed in chloroplast. So that leaves us with N-methyl transferase needing chloroplast C to 4 and tabersonin 16 hydroxylase needing external phase of ER A to 2. So C4 A2, which is option A, and the remaining two enzymes can function in cytosol. Okay, right. Tanmoy, we have covered most of the questions, but uh, mm, if you have any doubts, you can ask once we cover the other questions as well. Okay. So the next question is to reconstitute the alkaloid indole alkaloids in Nicotiana benthamina, scientists transformed the said plant with PAS, DPAS, and tabersonin synthase genes. Upon feeding stemmadenine acetate in the transformed plants that co-express these three genes, which of the following metabolites cannot be formed? Dehydrosecodine, dihydroprecondylocarpine acetate, tabersonin, and catharanthine. So for this, we need to know where exactly stemmadenine acetate is getting converted to what and what these three enzymes take part in, what reaction these three enzymes take part in. You can think about this question for a couple of minutes and then we look at the uh, entire reaction pathway. Yeah. Do you guys want to try this? Any one of you? Yeah, you can unmute.
I think here tabersone and catharanthine both are found. Uh,
Hmm.
not being found. Dehydroxychloroquine.
So option B or
So
option you're saying option A, is it? So if you look at the pathway here, you have stemmadenine acetate. Now the enzymes that we have here are PAS, DPAS, and TS, right? So stemmadenine acetate gets converted to precondylocarpine acetate, which gives us dehydrosecodonin. Now here is where is the answer deciding step. If you have tabersonin synthase, from dehydrosecodonin you get tabersonin. If you have catharanthine synthase, you have catharanthine. Since we have only tabersonin synthase, tabersonin is produced and catharanthine cannot be formed. That is why option D is the right answer here. Okay, this step. Hmm. Right. The next question is identify the specific region of Catharanthus roseus leaf where the initiation and completion of windolin biosynthesis occur. And four different options are given here: palisade parenchyma and spongy mesophyll, respectively, epidermis and spongy mesophyll idioblast, respectively, epidermis and palisade parenchyma, respectively, palisade parenchyma and laticifier, respectively. So take a couple of minutes, think about where all these Windolin intermediates, precursors travel through, like we just discussed for specific organelles. Think about at the tissue level also, and then we'll get back to this question. Yeah, Tanma, you can unmute. Why do you think it's option B?
Sir, in the epidermis, first it will be synthesis initially from tryptamine, secologenin to tabarosin,
Hmm.
and sir, then it will be the rest process. It will go from the intermediate M1A to the spongy mesocell, idioblastic, and lenticular cells,
Okay.
where the windowlin will be synthesized.
Is there any specific reason? You're right. This is how the process looks like. Is there any specific reason for this
Sorry.
compat? No. So technically, all of these enzymes can function in the same tissue. But why do you think such compartmentalization is needed? Do you think is there any reason?
Sir, because of the chloroplast presence, that's why.
And how does chloroplast presence how do you think it would affect here? Okay, so this has been discussed in a PNAS paper. So here these enzymes are, which is D4H and DAT, they are present in idioblasts. So how one can understand this is in tissues, not all genes are expressed at all times. Oh, here, if you see, you have upper or lower epidermis here, the cuticle, which is above the upper epidermis. Then you have idioblast. Now, this below this idioblast, you also have the lattice of fire. Now, because different sets of genes are expressed in different tissues, you have a requirement for the transport of the metabolites. If all genes are expressed within the cytosol, then you cannot control the rate of synthesis of specialized metabolites. As you see here, you have a common precursor that gives rise to tabersonin and catharanthin as well. And if you want to differentiate the pathway, like how it is done here, some parts of windowlin are going into this lattice of fire, whereas those that are going to catharanthin are going to the upper cuticle. So you want this differentiation of pathway. How can you do this differentiation of pathway? By expressing specific genes in specific tissues only. That is why this pathway here, you see that specific genes are happening in specific tissues. Was that clear?
Yes, sir.
Right. Yeah. Okay. So with this, we come to an end of this week's lecture. If there are any questions with respect to the lectures this week, you can ask me. I'll stay on the call.